It's my distinct pleasure today to introduce Colin Pritchard, our very own Colin Pritchard. He's a professor now in the DLMP. So Colin actually is one of the people that has known no other institution other than the University of Washington. He started went his undergrad here and double majored in biochemistry and philosophy. I did not know about the philosophy thing. And then he went through the MSTP program and then moved on up through the ranks, starting in laboratory medicine, and is now a full professor in DLMP. He's also the program director for cancer precision diagnostics at the Brotman Baty Institute, which I think he'll touch on a fair amount today. Colin is really well known in the cancer testing field with a heavy emphasis on like prostate cancer. He's got a large number of grants and stuff like that focused on prostate cancer and the tumor genetics associated with that. One of the big reasons I wanted to have Colin give a talk is because of this new tumor sequencing initiative that's been started. And I think we can all learn a lot from this, and I'm hoping to hear some really interesting stuff here. So take it away, Colin.
Thanks, Scott, for that very kind introduction. I just want to say it's really an honor and a pleasure to be able to talk to the department. I give a lot of talks, but I always value giving a talk to my colleagues the most. So I've tried to sort of call out people throughout. I'm sure I've forgotten people though, and I apologize in advance for any of those oversights. But what I'm going to do today is give a very kind of high level talk on precision oncology testing, so cancer testing, where we are at in 2023. And as Scott kind of alluded to, what it's going to take to go big. And if we want to be doing precision oncology on most or all of our cancer patients with life-threatening cancer. So I have no relevant disclosures. So here are the learning objectives. You can briefly peruse these. So when we're thinking about precision oncology testing, at a very high level, we can think about different reasons for testing. So we can test for cancer risk, looking for inherited variants. We can test for treatment decision making, which is, I think, where a lot of the bulk of precision oncology is right now. Test the tumor, see if there's a genetic mutation, make a treatment decision. We can do treatment monitoring, especially in the setting of cell-free DNA, where it's much easier to get access to tumor DNA. We can test for prognosis. And of course, we've been doing this for a couple hundred years in pathology. We can test for diagnosis. This talk, particularly the first part of this talk, I'm going to focus on areas of cancer genetic testing where there's interplay between tumor and germline. This is something we specialize in the department and the division in. There are roles for paired testing in targeted therapy. There's roles for paired testing in looking for inherited cancer syndromes and a cancer syndrome workup. And there's roles for paired testing in cell-free DNA, or also known as liquid biopsy. I'm going to focus in the first part of the talk on DNA repair mutations in precision oncology because it's what I happen to work on. But it's also something that I think is really central to a lot of the precision oncology testing we're doing right now, both in the treatment setting, but also in the cancer risk setting. I want to sort of preface beforehand, though, this is by no means the breadth of everything in precision oncology. We have to sort of narrow it down. So I'll talk about some background on DNA repair genes. What are the pathways? What are the genes we test for? What's the clinical utility? Some testing approaches, some of the assays that we offer, and then some of the roles for paired tumor and germline testing for these genes. And then in the second part of the talk, I'm going to talk about some of the emerging tests that we have in cancer precision oncology. Things like polygenic risk scores, digital pathology and artificial intelligence, and a few others. We'll just give a little bit of a flavor of sort of what's to come. And then at the end, I'll touch on some of this sort of scaling issues. So if we're going to have precision oncology for all, what's it going to take? What are the steps to make that happen? So DNA repair genes, there's really two main pathways we think about with DNA repair genes in cancer testing. And those are the homologous recombination DNA repair pathway and the mismatch repair pathway. And so homologous recombination DNA repair, best known for BRCA1 and BRCA2 genes. There's a germline syndrome associated with breast, prostate, pancreatic risk, the King syndrome. And there's treatment implications if you have either an inherited or acquired mutation in one of these pathway genes for a type of drug called a PARP inhibitor, and also tends to sensitize tumors to platinum-based chemotherapy. For mismatch repair, there are some example genes here, the MSH2 and MLH1. There's a few others. There's a well-characterized germline syndrome associated with colon cancer risk, endometrial cancer risk, prostate cancer risk, and others called the Lynch syndrome. And if you have either inherited or acquired mutations in this pathway gene, you tend to be responsive to a kind of immunotherapy called checkpoint blockade immunotherapy. So these are actionable in a number of ways. So, gosh, going back a few years now, we were lucky to be involved in a large multi-institutional study looking at the prevalence of inherited DNA repair mutations in prostate cancer. And at that time, it was thought that this was very rare. But what I think hadn't been done is looking specifically in metastatic prostate cancer. If you look in and that's the lethal form. Fortunately, most men with prostate cancer don't die of their disease. They just, they're cured or they have localized disease only. But the approximately 10% of men who do get lethal disease, if you focus on those men, you see a very high prevalence of inherited DNA repair mutations, particularly in BRCA2, but also in these other genes as shown. And when you look in the tumors, you see there's evidence that the second allele is inactivated. So those of you remember the Knudsen two-hit hypotheses, these are tumor suppressor genes. So if you're born with one defective copy, you really want to see that second copy of the gene inactivated in the tumor if that gene that you inherited the deleterious mutation in had anything to do with the tumor. And that's exactly what we saw. So, and we're seeing this again and again, not just us, but many others, that these inherited DNA repair mutations are much more common than we thought in many cancer types. So I've used this term King syndrome to describe men and women with inherited BRCA1 and BRCA2 mutations. And the reason that I favor this term over the more conventional hereditary breast and ovarian cancer syndrome term is that it leaves out things like prostate cancer and pancreatic cancer, and that it implies that only women are at risk for cancers just by the name. So why not name this after Mary Claire King, who discovered the BRCA1 locus, just like the way, and described some of the first families, just like the way we've named Lynch syndrome after Henry Lynch, who did very similar thing for mismatch repair. So even Li-Fraumeni syndrome, this is the inherited cancer syndrome caused by inherited P53 or TP53 mutations. It's a very rare but severe cancer risk syndrome. Even this is more common than we thought in older adults. So we did a study that was just published last year in collaboration with Kara Maxwell at Pennsylvania, and Heather Chang here at the Hutch, where we looked at a number of cohorts of men with prostate cancer, and we also then looked at a number of Li-Fraumeni cohorts of families known to have germline P53. And what we saw is almost a 25-fold incidence of prostate cancer. And surprisingly, about 1 in 200 men with prostate cancer had a germline P53, which for this very rare syndrome is about 9 times higher than the population. So, and what we saw, not surprisingly, was that these variants tended to be not classical Li-Fraumeni syndrome variants that cause very severe pediatric cancer, but what we call attenuated variants. So the cancer's risk is not as extreme, but it's still a very important cancer risk. So you basically can live long enough to get prostate cancer, because prostate cancer is a cancer of older adults. And so this is just an example. We're seeing this time and time again for even these very well studied genes that, gosh, maybe the story is a little bit more than we thought. This is a busy slide, and the point of the slide is just to say that not all DNA repair genes are the same. There are some FDA approvals that are sort of lumped together, where a lot of genes together, if you have a mutation in any one of these genes, you can be eligible for, say, a PARP inhibitor. Or if you have a mutation in one of these genes, you may be eligible for an immunotherapy. But the reality is that the genes behave very differently. And it is important for us as molecular pathologists to really understand this subtlety and help our colleagues, particularly in medical oncology, to understand this as well. Okay, so I'm going to switch gears now to testing approaches and some of the testing we do here. But again, with more or less a focus on DNA repair genes and precision oncology. So if we think about broadly what clinical testing is, we have 3 stages, right? We have a pre-analytical stage, that's the specimen. We have an analytical stage, that's we thought of as the assay. And then we have a post-analytical stage, which is the analysis. And I think that the reason I'm emphasizing this, of course, I'm talking to a lab medicine and pathology audience, and most of us understand this. But we are somewhat unique in that way. I think a lot of the field thinks of these tests as these black boxes, and they're focusing really more on this analytical stage and sort of neglecting the pre-analytical and post-analytical stages. And so it is really important to think of all 3. And I'll get to some of those points in a minute. So we've been very lucky to have broad support from the department and been able to implement many cancer next generation sequencing tests over the years. The point of this slide is to show kind of a timeline of different tests that have been implemented, and also to sort of highlight that it's not a one size fits all. There really are different tests that are useful for different indications. And the other point of this slide is to highlight that there are many applications of paired germline and tumor, or germline and somatic. I'm using somatic for tumor here. And so I'll get to some of those coming up. So one of our workhorse tests is our comprehensive cancer sequencing panel, the Oncoplex panel. And we also do this on cell-free DNA, and we call that Oncoplex CT for cell-free tumor DNA. This is currently about 2 million bases of DNA sequence. It's a little over 350 genes. Importantly, it's designed to detect all classes of mutations. And so what I mean by that is single nucleotide mutations, small insertions and deletions, copy number changes in the genome, gene fusions, microsatellite instability, and I'll highlight that work coming up, tumor mutation burden, most recently a signature of homologous recombination DNA repair deficiency, and also most importantly, I'd say, is this patient-tailored expert interpretation aspect. And again, that's where it's not just about what the test does; it's about how the test is interpreted. So if we focus on the pre-analytical phase of precision oncology testing, I mean, this is absolutely critical to getting the answer right. So it's garbage in, garbage out. Pathologist review is critical. So in the genetics division, we have section head Eric Connick, and who's got a team of faculty and staff, including Regina Kwan, and Anshu and Vera Paulson, who just do an amazing job sort of coordinating the specimens that come in. So what they're doing is they're looking at slides, they're making sure that there's adequate tumor content, they're actually circling areas of tumor so that those areas can be scraped off and enriched when we do the tumor-based tests. But they do quite a bit more. There's also new kinds of specimens like cerebral spinal fluid or ascites fluid, of course, cell-free DNA. And so this is really an emerging subspecialty area. We call this the Genetics Pre-Analytical Services section, or GPS section. And I wouldn't be surprised if in some time in the future we might have like a pathology subspecialty fellowship in GPS pathology. It's becoming that critical, this pre-analytical component. So now I want to switch gears to some of the post-analytical pieces and some of the mutation signature biomarkers that you can get from this kinds of panel tests like Oncoplex and similar. So I mentioned already the homologous recombination DNA repair deficiency signature. This is the signature of BRCA-ness. That is, if you've got deficiency in that pathway, there's ways to measure that. And if you have that signature in certain tumor types, that might predict PARP inhibitor therapy. Microsatellite instability is another thing you can measure from next generation sequencing. This is the mutation signature of mismatched DNA repair. And if you see that signature, it's pretty good indication you have mismatched DNA repair deficiency, and then you would be eligible for immunotherapy. One of the newest signatures that is used clinically right now is tumor mutation burden. And so it's a fairly simple idea, just count up the total number of mutations you see per a certain unit of DNA. It's usually typically done in a total number of mutations per megabase or million base pairs. And that's a surrogate of the tumor antigen load, and predicts immunotherapy for that reason. So I mentioned this homologous recombination deficiency signature of BRCA-ness. So work led by Steve Salaponte and Nick Crum in our NGS analytics and genetics divisions in our department has really developed really cutting edge technology to be able to measure the signature. It uses paired tumor normal testing, and the core of the assay right now that Steve and Nick have developed is really focused on loss of heterozygosity across the genome. So when you have homologous recombination DNA repair deficiency, you get losses of whole chromosome arms, that kind of thing, and that's measured as increase in global or genomic loss of heterozygosity. Also want to highlight the work of Grogan on the team and many others in the NGS analytics division who have really, spent a huge amount of effort to develop this test. In addition to the loss of heterozygosity, you can look for things like the rescue pathway signature, which is non-homologous in-joining signature. That rescue pathway comes in when you have deficient homologous recombination repair, and that is manifested by these mutations in these areas, these little deletions in areas of microhomology. There's also a large rearrangement genomic signature. There's a single nucleotide signature. And so in the next iterations of these assays, I think these will continue to get better and better as we incorporate more of these hallmarks of a homologous recombination DNA repair deficiency. But again, the point of these is primarily to predict PARP inhibitor responsiveness in tumors. And again, this is just output from the test. What we're showing here on the top is an example of an LOH or loss of heterozygosity low tumor, basically negative for the signature, and then on the bottom would be in loss of heterozygosity high tumor, which would be positive for the signature. Again, work led by Steve Salaponte and Nick Crum here, shifting gears now to another signature, so microsatellite instability, my next generation sequencing. So this is something that is a little more mature, and it can be done very well. There's a number of methods. Again, Steve Salaponte developed a method that we collaborated on, going back 10 years ago now, called the MSINGS method for MSI by NGS. And it just works really, really well, especially in tumor types that are not the standard tumor types that MSI testing is done. So for those of you who aren't really experts in this field, MSI is a test that we've been doing by other methods for decades, but we've primarily been doing it in colorectal cancer and to a lesser extent in endometrial cancer. And it turns out that those traditional methods that use PCR and capillary electrophoresis that only look at a few microsatellite markers and are kind of have sparse data just don't tend to work as well in new cancer types. Whereas the NGS-based methods, where you can get lots of microsatellites and you get more robust data, you can also focus on microsatellites that might be a little bit more better sensors for that tumor type, work better. And so this is an example of data, in this case from prostate cancer, showing that traditional methods of MSI are only about 70% sensitive in prostate cancer, where this MSI by NGS method was about 95% sensitive. And this is work led by Jennifer Hempelman, Steve, and others. So the last mutation signature I want to highlight is tumor mutation burden. And in this case, we use tumor mutation burden, again, to remind people as a surrogate for the antigen load, and therefore as a surrogate that the tumor might respond to immunotherapy. So if you have a high mutation burden, that means you can get immunotherapy, and that's an FDA-approved indication as of 2020. But here on this slide, what I want to highlight is the pitfalls of this biomarker. So there are many false positives in non-European ancestry, and that's thought to be because what we're doing when we're doing a tumor-only test is we're capturing both the germline and the tumor. And if you have rare germline variants that are underrepresented in population databases, those are going to be miscalled as mutations and falsely elevate the tumor mutation burden. So it's really a big problem. Tumor mutation burden is also affected by specimen and sequencing conditions, and methods vary between labs. So we've argued that really automated tumor mutation burden is not enough. It's not reliable. This test really should be thought of as sort of a two-step process. There's an automated component, and there's a pathologist review component. Very similar to the way we think about an immunohistochemistry test or a fluorescence in situ hybridization test. We know that there's going to be batch artifacts and things that a pathologist's eye can sort out very quickly, but a computer still has some trouble with. And it's no different with tumor mutation burden. And because this is being used as a sole biomarker for very high-risk decision-making, it's probably worth doing. And so work led by Regina Kwan, highlighted here, is looking into this question and following up on this nice study that we weren't involved in that was just published in Cancer Cell less than a year ago, showing that this problem of ancestry bias or ancestry interference is so bad that tumor mutation burden as a biomarker doesn't even really work for immunotherapy prediction if you have African or East Asian ancestry. It only really works well in European ancestry. Now, again, that's using automated commercial lab tumor mutation burden. That's not using the kind that we're advocating for, which is basically a pathologist gets involved and makes a call. So that's work Regina's actively working on, and we're hopefully will really improve this biomarker. The other thing that's really important in the sort of post-analytical phase of precision oncology testing is the interpretation of the variants. And it might seem like this is mostly sorted out in 2023, but it's not really. We still know very little about the genome, and we still know very little about how to interpret genetic variants. So this work published a few years ago, led by Jen Dines, basically highlights that in BRCA1 and BRCA2, some of the best studied genes we have in genetics, we're still not doing very well with our variant interpretation. And what I mean by that is we know a lot about this gene. We know that the middle section of BRCA1 and the middle section of BRCA2, about 60% or more of each gene, it's like a giant intron. It's not important. It tolerates any kind of variation. You can cut that section out of the gene and put the C-terminal and N-terminal together, and the protein will be perfectly functional. Moreover, there's never been a single pathogenic variant that's a missense variant, that's a protein coding variant, that's ever been described in this huge chunk of the gene. Despite that, almost all commercial labs are still calling rare variants, rare missense variants in these regions of the genes as uncertain significance, and they are clinically reporting those back to patients. And a surprisingly, I would say shockingly high percentage of the time, there's now data we're seeing those patients are getting prophylactic mastectomies, irreversible surgeries on the basis of this misclassification. So the risk for harm here is not only real, it's extreme. And so we really, I think, need to do a better job with variant interpretation. This is just one example. So what we did following up on this paper, and I give credit to one of our talented staff scientists, Bridget Kurosukawa here, for this idea, which is an obvious idea, but not one that we had thought of, is just why not just submit every single missense variant, that is protein coding variant, that's in these quote cold spot regions of BRCA1 and 2 that we've defined that we know are likely benign. Why not just submit all that information to this clearinghouse of variant interpretations called ClinVar? And that might help the whole field. And so that's what we've done. And what I'm showing here is that, there was over 6,000 interpretations submitted. And what that did is it changed over 5,000 interpretations in BRCA1 and 2 from a variant of uncertain significance interpretation to a quote conflicting interpretation, because we're an outlier calling it likely benign. But we're hopeful that this might prevent some of these iatrogenic harms like unnecessary surgery that we know are being caused by variants of uncertain significance. So another really important part of interpretation is a group approach. And when we think about 21st century genomic medicine, it really ought to be patient tailored. And it really, I think it demands multidisciplinary or at least multi-team review in order to really get it right, at least for these very complex tests. And so we're very lucky to have a very talented team that we call the Clinical Genomics Analysis Team or CGAT team, highlighted here with our leads, Ankita Jirani and Angela Jacobson. And we have a very talented team of directors in our department too. And so the way we do all of these complex tests is we get the sequencing data that the NGS analytics team does such an amazing job with their pipeline packaging up. And then we have this team of variant review scientists and lab-based genetic counselors who are able to sort of do an analysis of this data and even draft reports. Then we have the pathologists and other lab directors review the data individually and then in typically weekly or sometimes even bi-weekly consensus review conferences before there's a report. In addition to that, there's monthly molecular tumor boards that are disease specific. So this, I think, is really an integral part. And there's just so many cases where it comes to what we call sign out, the multiple director review stage. And somebody has a really insightful comment that really changes the way you think about something and really enhances that report for the patient. So for molecular tumor boards, as I mentioned, we do these as a disease specific. We now have the prostate or genitourinary board that's been going the longest, 10 years now that I lead. We have a GI, gastrointestinal tumor board led by Dr. Connick, a pediatric tumor board led by Dr. Lockwood, breast and Dr. Paulson, and breast led by Dr. Connick, and then gyne as well led by Dr. Paulson and Dr. Lockwood. So we have multiple of our somatic or tumor-based directors leading these different tumor boards. We keep proliferating these. At some point, we might want to go to like a more frequent but pan-cancer tumor board format. We've been talking about that for a while. We'll see where that goes. But it's a great resource for talking with our colleagues and getting input from the clinicians who are ordering the tests. Okay, so in the last part of this first section, I'm going to talk about roles for paired tumor germline testing. So I'm going to circle back to Lynch syndrome, and just to remind people again, Lynch syndrome, it's an inherited syndrome when you have inherited mutations in mismatch repair gene pathway. It's fairly common, as much as 1 in 300 people. And if you have colorectal cancer, about 3% of people with colorectal cancer will have Lynch syndrome. For this reason, universal tumor-based screening is recommended to look for these inherited mismatch repair mutations in colorectal cancer and also endometrial cancers coming. So as a result of universal screening for Lynch syndrome, what we and others have found is that it turns out that only about half the time when you see a positive MSI result in Lynch syndrome screening that is not explained by the common non-inherited cause, which is MLH1 methylation, when that's been ruled out. Only about half the time it's Lynch and half the time it's this flavor we call double somatic, meaning basically you just got both mutations in the gene happened in the tumor. And because historically a positive MSI result for Lynch syndrome screening in this context that was not explained by MLH1 methylation was considered to be Lynch syndrome until proven otherwise, there's now clinical utility to actually understanding what is the mechanism of my MSI. And so that's what we've done. We have a test called Colin C tumor, which is a paired test where we look at the germline in the tumor. And these are typically patients with colorectal or endometrial cancer that have had a positive MSI type screening test and yet have had no germline variant identified. And the clinicians worried like, well, gosh, was the germline variant just missed? Does this patient really have Lynch syndrome and we just missed it? We'll feel much more reassured if you show us from the tumor that we know why that screening test was positive. And so we do that, and in that clinical scenario, about 75% of the time it is solved as double somatic. But importantly, we do find some missed Lynch syndrome cases about 7% of the time. We do find some missed amyloid methylation, some false positive immunohistochemistry, et cetera. And this is work led by Drs. Connick and Angie Jacobson and Brian Schertz that's in preparation for our clinical experience. We've been doing this test for, gosh, almost 10 years now and have just a really nice wealth of data on it. Another newer test that employs kind of a similar principle is what we call the Colon-C polyposis test. And this is a 3-sample test. So now we're taking 2 neoplastic samples and 1 germline sample. What we're doing here is we're taking a patient that has many colon polyps. And typically they've been worked up for one of the germline polyposis syndromes like familial adenomatous polyposis caused by mutations in the APC gene, and no mutation in APC has been found in the blood. And so then the question is, well, is the patient constitution a mosaic? Is the mutation in, their colon and maybe other tissues in their body, but just not in the blood? And so how do we prove that? Well, we have access to distinct neoplasms. We can take 2 distinct polyps that are distinct neoplasms and the germline, and then we can see, do 2 distinct polyps share the same APC mutation? If so, that's pretty strong evidence that they have a mosaic APC syndrome, a mosaic familial adenomatous polyposis syndrome. And this over about 2/3 of the time, we do find mosaic explanation, or at least we solve them one way or the other in this highly selected scenario. And this is work led by Dr. Eric Connick and Andrew Jacobson. It's really a nice, unique test that we offer. You can also use paired tumor NGS to do germline variant interpretation. So particularly for patients with germline mismatch repair variants of uncertain significance, abbreviated MMR-VUS here, you can use what we know about tumor mutation signatures, what we know about expected second hit for the Knudsen 2-hit hypothesis. You can use this kind of reasoning to look at the tumor and then potentially reclassify a germline variant as pathogenic or benign. This is work led by Brian Schertz and Angie Jacobson, and we've seen that up to 80% of the time when we strongly suspect a variant might be pathogenic, we can reclassify it this way. Okay, so shifting gears to another area where I think paired tumor and germline testing is important is liquid biopsy or cell-free DNA. So let's just take a step back. What is this? So we're looking for shed fragments of DNA into the plasma from a tumor. We're then measuring those, sequencing those fragments to then make inferences about the patient's cancer. So the strengths are, it's a non-invasive. You can sample all metastatic sites because they're all dumping into the plasma. You can fairly easily do serial monitoring of disease, but there's fairly important pitfalls. So there's not standardization on tumor content or clinical features. Targetable mutations can be in a subclone that maybe you're just targeting one of the metastatic sites and not all of them. And clinical false positives are a problem. So this is work that we did with Mike Schweitzer a few years back, where we looked at the performance of liquid biopsy, in this case in prostate cancer, on the basis of a surrogate of disease burden, in this case, the PSA test. So in metastatic prostate cancer patients that had higher PSA greater than 10, when you did a liquid biopsy, the cell-free DNA mutations you saw in plasma matched what you saw in the tumor. However, when you had lower burden disease, less than 10, you saw almost no match. And the mutations you saw in plasma were almost entirely clonal hematopoiesis. So what is clonal hematopoiesis? It's also sometimes called clonal hematopoiesis of indeterminate potential. This is something that happens in your white blood cells as we age. And you get mutations that are selected and form these little clonal populations of white blood cells. And you can get mutations in genes like TP53, ATM, CHEK2, even BRCA2. And so some of these same genes that are associated with clonal hematopoiesis are the same ones we're testing for to make treatment decisions. And inconveniently, these clonal hematopoietic mutations are also induced by cytotoxic chemotherapy. This is a couple of studies published back-to-back, one led by Liz Swisher, one by another group, showing that this is true for ovarian cancer, and this has also been shown to be true for other cancer types. And this is work from out of the Hopkins showing that there's very poor congruence in commercial liquid biopsy tests between labs. And I think part of this issue is this clonal hematopoiesis. So with Kendall Jensen, one of our molecular genetic pathology fellows a couple of years ago, we decided to look at this problem for prostate cancer. And what we had been doing is paired plasma and blood testing from the beginning. So we know with certainty when there's a clonal hematopoiesis mutation, we know with certainty in our test because we'll see it in the white blood cell test control. So we're in a unique position to sort of say what the scope of this problem was. And unfortunately, it's really a major problem. So in prostate cancer, metastatic prostate cancer, about 10% of the time, if you get a commercial cell-free DNA test, you're going to get a clinical false positive result that's going to tell you to use something like a PARP inhibitor on the basis of a clonal hematopoiesis mutation. This is a major problem. I mean, that's a major diagnostic error with major consequences. And so what we've argued is that the solution to this is when you're doing a cell-free DNA test, a liquid biopsy test, just have a paired blood control. It couldn't be easier. You can use the same specimen to make your control and your plasma sample. It does add some cost. It adds a little bit of logistical complexity, but we're hoping that's where the field will go with this paired testing in ctDNA to really improve the performance by eliminating this interference. Okay, so now shifting gears, I'll talk about some emerging tests in precision oncology. I'm just going to have a very brief overview here. So one of the emerging tests is polygenic risk score. So what are we talking about there? So we're talking about relatively common variants that are inherited variants. We're not talking about tumor variants. We're talking about germline inherited variants. You measure multiple of these variants. You come up with an aggregate score that says I'm at higher risk of this cancer or that cancer. And there have been multiple of these large genome-wide association studies that have, proposed some of these risk variants. And we're getting better at this. There's a breast, there's a prostate, there's a colon, PRS. We've built these in to the backbone of our cancer risk test, which is called BRCA. We're not going to be, I want to emphasize though, we are not clinically reporting this now because it's not clinically recommended. But I think it is the time to start gathering this data so that we can prospectively validate polygenic risk scores to see if they really work or not. And we're doing this as part of the PATROL trial, which is a screening trial for prostate cancer in men with high genetic risk, men with known inherited BRCA2 or similar mutations. This trial has been going on for a few years, led by Heather Chang, Dan Lin and Virtue Darst, and Saji Hassan and Bridget Kurosawa and our group are getting involved now in this polygenic risk score aspect. Bridget played a big role working with Virtue and myself in kind of designing this. And then Saji is going to be doing coordinating the interpretation and sequencing. So we're very excited to kind of get into this area and see if it really works. But again, we're not going to be clinically reporting this, and we're going to be doing this as a post hoc analysis on a research-only basis as a part of this trial, just to sort of get our feet wet, so to speak. Another up-and-coming area with regard to cancer early detection and diagnosis are what's called multi-cancer early detection tests, also known as MCED tests. These are emerging. There's one commercial option out there that's live now. It's not recommended clinically by any guidelines, but it is available. And what these tests are doing is trying to actually look for trace amounts of cell-free DNA in the plasma as a way to do cancer early detection. This is devilishly hard to do, but the tests are getting better. They're taking advantage of methylation patterns that are kind of specific, like a methylation barcode pattern that you can look at on a molecule by molecule basis, and taking advantage of so-called fragmentomics, like the size of cell-free DNA fragments, and then where they map on the genome. You can make inferences about the actual expression and the cell types they come from. It's pretty remarkable stuff. With this same kind of technology that MCED tests use, I think we could actually get to the point of true liquid biopsy. That is a true diagnostic cell-free DNA test that might mean you don't even need to get a tissue diagnosis. Now, a lot of anatomic pathologists on this call are probably going to be like, no way, we're never going to do that. But I think the technology is going to get there. I don't mean that tissue diagnosis is going to go away across the board. Probably not even going to go away in a large number of places, but I think we can get there. And this is work that Adil Mohammed, who was with us as a master's lab ed master's student, is now with Gavin Ha and Michael Hafner as a PhD student is working on. There is a multi-cancer early detection working group that was just fired up, led by Marianne Dubar-Galt and Ruth Etzioni at the Hutch. I'm lucky to be a part of with these other folks, and I think the goal of this group is to be inclusive. If other people are interested in this, reach out to me or some of these other people if you want to get involved. It's just started, but the idea is to sort of facilitate informed uses of these tests. Like I said, there is a commercially available test. These tests are going to start to be used. We're going to, as laboratory medicine and pathology physicians, we're going to be asked to really interpret these for patients. It's a coming storm. And the idea is to bring in testing into our system appropriately through clinical trials and through evidence base. Okay, and then on the diagnosis front, this is possible from cell-free DNA, as I mentioned, by inferring gene regulation patterns and nucleosome phasing, and work led by Gavin Ha at the Hutch and Navinal Sarkar, and again, Adil Mohammed and others has really shown that you can basically subtype prostate cancer into neuroendocrine-based prostate cancer or androgen receptor-driven prostate cancer solely on the basis of a cell-free DNA test, which is just remarkable. Okay, so digital pathology and artificial intelligence. Couple slides on this. This is obviously going to be important for the future. Dr. Baird and others have identified this as a priority for the department in terms of the direction. And it is really an exciting area. So how does digital pathology and AI fit in with precision oncology and molecular testing? Well, work led by Michael Hafner and Stephanie Harmon is really making this happen. So Michael Hafner is in our department, a GU pathologist and extremely talented researcher, and Stephanie Harmon is a computational biologist at the National Cancer Institute. And together they've working in this artificial intelligence resource that I'm lucky to be a part of with these other people. And what they're doing is trying to train models of scan slides, integrate that with molecular findings like Oncoplex data to do outcome prediction. And so we've been doing this pilot study with Michael Hafner, Eric Connick, and our trainee Heather Kirkham for the last couple of years. What we're doing with our prostate cancer cases is the prostate cancer cases that go for Oncoplex, Michael's lab then scans, and then we can use these trained models to do this data integration. So we're very excited to see where this is going. It's still early days, but we have several hundred cases now scanned and annotated in a structured way. The other thing I think is coming is whole genome tumor sequencing. So, right now we're kind of in this panel phase with Oncoplex, but really the end game, I think, in precision oncology, at least for DNA, is going to be whole genome. And we have a pilot study set up in collaboration with the Hutch, with Eric Holland and his team, and then Saji is on, Dr. Connick, Chaz Quinn and Sarah Trignano on our team are involved in this very early pilot where we're looking at paired Oncoplex data and then seeing how whole genome sequencing can do. We also are going to do whole genome transcriptomics with RNA-seq. So again, this is just a pilot. We're not going to be reporting these back to patients, but it'll be exciting to see what happens. Okay, so in the last 10 minutes, I want to talk about scaling. So looking ahead, if we're thinking about the field of precision oncology, I think it's useful to really take a historical perspective. So let's look at radiology, another diagnostic field. So you could argue that radiology really first came on the scene in 1895 when a key technology was invented, X-ray. And I'm gonna designate next generation sequencing technology, which was invented in 2005, as our X-ray. Okay, so if you look at the parallel timelines, we're in 2023, so we're in about 1915 in terms of radiology timeline. The first radiology residencies weren't until the late 20s. Interventional radiology wasn't until the 1960s. We didn't get CT and MRI until the 70s. So I think it's useful to think about genomic medicine and precision oncology as really an early emerging field, but I think it's really ripe to take off. And I don't think it's crazy at all to think that we'll have an interventional genomic medicine residency with learning how to do CRISPR interventions or whatever the newest thing is in 50 years down the road. And thinking in this way, I think, is useful when we're thinking about going big. So what's part of the other rationale? You know, we want to improve patient care. We want to position our institutions for this future that's coming, and we want to be equitable. Right now, you know, there is inequities. These are expensive tests, but also we want to think about research. We can't really tease apart what we're doing from research. It's very early days. I mean, we're learning as a field as we go. So I think it's part of the mandate for our clinical testing programs is to have a research component built in so we can take advantage of what we're learning to improve care. So we have this tumor sequencing initiative that Scott mentioned at the beginning, and the goal is to sequence, and this is you know, Tom Lynch, the president of the Hutch, has really made this clear with his vision, to sequence all life-threatening tumors in system, make data readily available for research. And the goal is to use Oncoplex plus research testing. Again, the whole genome sequencing will probably play a role here. Create a data portal, probably going to be this data portal called CBio, but it also might include other visualization tools. We're already seeing this rapid expansion in genetics test volume. You can see just in the last couple of years, things are really starting to take off. And a large part of this is due to next generation sequencing volumes. And with the recent integration of genetics division labs, this doesn't even capture the full volume. This is actually just a subset of division labs. So the expansion here is even higher than probably is being shown here. So we're already getting in front of this. This is really credit to our division head, Tina Lockwood, who's really an operational, has operational excellence, I should say, in helping us scale up for this sort of coming tsunami. Doing things like preparing for twice weekly runs, more instrumentation, many other things. I've been working with Noah Klemfus, who's the administrative director of the Brotman Baty Institute, with many meetings and sort of planning and thinking how we're going to go big. So to boil this all down of what we're working on, there's sort of 4 key areas. Research consent infrastructure. If you really want to make data available for research, you're going to have patient consent. Increasing operational capacity that we're again doing in close partnership with Dr. Lockwood and others. Standard work pathways for billing, which I'll talk about in the next slide just briefly. And the data portal, which I alluded to. We need to have this data be available both for clinicians and researchers to be able to use. So on the data portal side, we were lucky to get some BBI funding, and we did a pilot study. This is work with myself in collaboration with Alice Berger at the Hutch, also in collaboration with Nick Crum, Sarah Trignano, and others. And we learned a lot from this. We were able to get a local CBio portal instance. This is a data portal. A lot more is going to need to be done if we're going to bake this in to clinical pipelines. But I think we've learned a lot from it, and I think we can do this. As far as standard work pathways and billing, we've really taken a quantum leap with computerized physician order entry, CPOE ordering, and Epic and pathway. So work led by Regina Kwan, Eric Connick, Tina Lockwood, Nick Crum, and more recently Jen Greenwood in our division has really upped our game here in terms of making this easier for oncologists to order. Okay, I've got a colorectal cancer, I order this pathway. I have a lung cancer, I order this pathway, and making kind of more standard work that facilitates both ease of ordering, good clinical medicine, and facilitates streamlined billing. And so we're lucky to also have within our department the genetics pre-authorization and billing team, the GPAB team shown here with the folks listed here who are continue to grow and are really a key part of making this ramp up successful. The other newest team that is just forming, we're calling the Precision Oncology Support Team. And the idea of this team is that before we have a functioning data portal, what can we do in the meantime to help facilitate clinical trial accrual, to help with translational researchers? And so we have a team of research coordinators who are able to interface with our internal databases, get the genomics data that are needed, for example, for the phase 1 clinical trial program or other programs at the Hutch, in a structured, streamlined way, in a formal mechanism where we can capture all the pieces of information we need, like IRB approval, et cetera. And so we have Sarah Trignano, we have Jennifer Hempelman involved in this. We have most recently, Rachel Osborne is coming to our group. We're lucky to have recruited, and then Chaz Quinn supervising this group. So we hope that this team can really help kind of bridge this time where we don't have the greatest data portal, but we're hoping to get there. Okay, so I've talked about a lot of things, and to summarize, where are we with precision oncology testing in 2023? So I talked about next generation sequencing testing of DNA repair genes that's increasingly needed to guide cancer treatment. I talked about paired tumor germline next generation sequencing tests, and some of the examples of where there's a use, like in a Lynch syndrome workup, or like to avoid interferences with clonal hematopoiesis, and liquid biopsy would be another example. I talked about some of the emerging tests in precision oncology that aren't really used yet or are not recommended yet, but are really the future. So on the risk side, polygenic risk scores. Early detection side, we have multi-cancer early detection tests. On the advanced cancer side, looking at whole tumor genome sequencing and RNA-seq, and then also digital molecular and integration with artificial intelligence. And then at the end, I talked about scaling of precision oncology testing, which is really needed to, I think, meet this exponential growth in clinical need. And as you can see by our volumes, there really is an exponential growth. And I think it's going to continue to go that way. It's also needed for patient equity, and really is going to be important to enable research. So I'm so lucky to work with a large team of faculty. This is not even all the faculty. This is a subset of the faculty, and I'm sorry if I've forgotten someone on this slide, but it's really a pleasure to be able to work with so many talented people and such a large group doing clinical next generation sequencing services around cancer. And this is to highlight the division labs, particularly the genetics and solid tumors lab that I co-direct. They really do all of the work that I talked about. You know, all that oncoplex testing and those other NGS tests. We're just so lucky to work with so many talented people. And I tried to call out other people as I've gone. And so I think I'll stop there and hopefully have some time for some questions.
Thanks, Colin. Can you put up the last slide?
Oh, I'm sorry. Yes,
No problem.
let me do that.
So anyway, Colin is now open for questions. Larry,
Can you see the
Bruce,
slide?
go ahead.
Okay.
Yes.
Colin, this is Larry. Thank you for a wonderful summary of what you're doing, what's happened, and really presented in a manner I think I've learned a lot from it. So what do you envision with the fact that metastases from a given tumor can have different genomic abnormalities? I'm assuming some of those will be targetable and others won't, but let me know. And the fact that if they are different, one might want to actually sample a metastasis to which the patient is most vulnerable, but that might also be the hardest metastasis to sample.
Yeah, it's a great question, Larry. So, I mean, one of the advantages of the cell-free DNA approach is that you are going to potentially sample that heterogeneity between different metastatic sites. So we see that in real world setting too. And we know from work in prostate cancer in particular, but other cancers too, that there is significant heterogeneity between different metastatic sites. So right now, there's not really a clinical indication to like do a liquid biopsy and then, oh, let's now go after a specific metastatic site. But with some of the imaging technologies we have now that are sort of that between the genomics and the radiology, we may get there where we actually are doing that. But then it's not only just identifying what that metastasis is, it's also like, what is the clinical utility of just targeting one site, versus the whole cancer, and so much to learn there. It's a great question.
Yeah. Thanks again.
Go ahead, Rosanna.
Great, thank you so much. This was a great talk, Colin. I have a question about constitutional mosaicism. So you mentioned a little bit about it, so I was very excited to see that. It sits now like a new frontier, right, for cancer that is not inherited and is not sporadic. There is this mosaicism from embryonic development. So how do you see this coming into the clinic? What ideas do you have about testing for this?
Yeah, I mean, it is coming into the clinic. I mean, I think what we're going to see it initially is the kinds of clinical applications that we're doing now with patients that have kind of severe phenotypes, like they have a lawn of polyps in their colon, like 100 polyps that's not explained. And so that's where, clinically knowing about constitutional mosaicism is important. But I'll tell you an anecdote. I mean, we just had a case from a month ago or so, we were probably going to write up, where there was a very young patient in his 20s with rectal cancer and had a recurrence and had polyps but long story short, we think there might have just been a very, very short segment of constitutional mosaicism, maybe just in the hindgut, and so what do we call that? And it's still clinically important because he had a targetable mutation, in this case, a Pol E ultramediator mutation, that was shared in distinct neoplasms. So it's a great question. I mean, where we're going to go with this? I have this discussion with Dr. Mirza, Reda Mirza, who studies this in the pediatric side all the time too. Philosophically, what do we even call this constitutional mosaicism? What's the minimal unit? Like I talk about, if a kid has just a little teeny fetus of a finger that's overgrowth, is that an overgrowth syndrome or not? Because we all have some mosaicism. So there's some meaty stuff there to dig into for sure.
Thanks.
Any other questions? So I have a question, and this is more just, I guess, practical, and maybe I missed this in your talk, but the new tumor sequencing initiative that you're heading up, that is, what is that targeting? Is that targeting like whole exome, oncoplex, whole genome? I know you mentioned whole genome, but I didn't get the feeling that that's what the whole thing was.
Well, I mean, this is still in the planning phases, right? So I think the goal for right now is to really think about these broad categories and build up. And this needs to be in close partnership with the Hutch too, and thinking about the precision oncology initiatives there. So we're keeping a close eye on where all that's going. But we do want to sort of ramp up oncoplex testing for sure and have that be a core part, because we already do such high volume testing. It's such a key part. But what else is going to be added on to that? Where the data portal and the consents? It's still an evolving area. It's a multi-million-dollar problem, and we want to not a problem. It's a multi-million-dollar. It's a good thing, but we want to get it right. So it is evolving.
Any other questions? Well, all right. So I want to thank Colin again for giving such a great talk. It was absolutely fascinating. And also I want to kind of just remind everybody that, in fact, this is the last of the research and discovery seminars this year, and we'll reconvene next year. The idea is to do it live, so we'll actually have people here. Anyway, so thank you again, Colin, and hope you have a nice summer vacation.
Thanks everyone for joining.